Thank you very much. Thank you very much, Stan, for such a nice introduction. And I really appreciate that I have a chance to speak in this wonderful series, which I learned about just a year ago. And it's fantastic that I can join now as a speaker. So thank you very much for having me. So today I'll try to address the two major questions which we explore in our research, and that's how do cells respond to electromagnetic field and how and if they can generate electromagnetic field. So just to put you in a geography here, so I'm speaking to you from Czechia, that's short for Czech Republic, which is in the heart of the Europe. And our research team is part of Czech Academy of Sciences, which is institution, I would say, growing fast in the terms of quality of the research. And as a fun fact, we have the most intense laser in the world, which was built recently. So, well, we don't use it ourselves in our lab, but it's something which is interesting. I think we can be proud of. Particularly, my research team is physically housed at one of the 54 institutes. That's the Institute of Photonics and Electronics of the Czech Academy of Sciences. And we are quite small institute, totaling 100 people in FTE. And we are one of the six research units. So our team is bioelectrodynamics research team. Now, let me bring you to the final frontiers. So I guess that there are some fans of the Star Trek, which I learned recently. So I'm also a fan of science fiction. And I was always wondering if those of you are familiar, there's this funny device they use in this science fiction series of Star Trek. They call it tricorder. And it seems to be able to probe what's happening in organisms, in a human, well, in a body, and also capable of influencing and treating certain diseases. So to me, it seems to be like these kind of devices must be operating on some waves which penetrate and affect organisms. Here is a picture of a wave incoming to the cell, and probing the radiation or the field which comes out of the organism and probably carries out diagnostically relevant information. So this is what our research team is doing actually, and in order to address these questions, we have our people from different various backgrounds. Most of us are originally engineers from different backgrounds, but also we have biophysicists and physical chemists by original education. Our mission, as we formally phrase it, but it's something really we hold to, is advanced research of probing and influencing biomolecular systems using an electromagnetic field. Well, this mission is something we do on a long term. I guess we'll be doing as long as I will be leading the team. And where this mission should bring us, so what is our vision, is that this should lead to bring foundation for novel electromagnetic methods, which will enable gentler and more efficient bio-nanotechnology and medicine, hopefully not too far in the future. Well, currently the tools which we apply are advanced electromagnetic concepts, micro-nanotechnology enabled tools, and computational methods. So at first, I will address one area of the research. Well, that's how external electromagnetic field, particularly intense short electric pulses, interact with biomolecular and systems and organisms. And although this is like currently the major research area of our team, I will just spend just basically only a few slides compared to the latter part. So we understand that the organisms are complex. So we definitely do employ mechanistic understanding. We understand that the organisms are complex machines where individual components interact in order to make the whole thing work. However, we are also aware that mechanistic approach is not sufficient. We are trying also to approach the science integratively from a philosophical perspective. So we believe it's important to understand the parts, and equally important is also to understand the interactions between the parts in order to understand the whole. So in our team, we choose particular favorite part of organisms. So, and that's mostly tubulin and microtubules. So, and when I translate this general understanding to the particular words, we are trying to understand how biomolecular components and their interaction between these components respond to electromagnetic field. So we work mostly, as I mentioned, with the protein structures as microtubules and tubulin and cells in our research team, in the experimental part of our research. So why microtubules? I guess many of you are familiar with the structures, but I would like to reiterate why they are so interesting. So microtubules have several important biological functions. They basically enable cell division. That's what makes them also a target of roughly 30% of the drugs in a cancer, I would say, cancer drug market. And so because they well, basically are very important to stop the cell division if it's needed in the oncological therapies. Then microtubules are also important in the intracellular transport and also in intracellular signaling. So they're interesting from this perspective, from a biological perspective. Furthermore, the tubulin, the subunit of microtubules, is a protein which has very high electric charge and dipole moment compared to other proteins. So that's what we guess renders microtubule and tubulin more susceptible to electric component of the electromagnetic field compared to other proteins. Well, furthermore, microtubules, as you might be aware, or if not, I'll tell you, are fascinating in that they are dynamic structures, and they maintain their dynamicity even out of the cell. So actually what you are seeing here, this artist representation of how microtubules polymerize or depolymerize, or people sometimes say this, they self-assemble and then disassemble. This can be reproduced in vitro out of the cell. So kind of we bring a little bit of non-equilibrium processes in the cell out of the cell, and still we can make it work. So what we do in our research in order to address how electromagnetic field affects biological systems, particularly microtubules, we typically start with molecular simulations. So mostly we do classical molecular dynamic simulations. So what you can see here is just a representative video of several biomolecules in the water box. With the blue arrow is the dipole moment of the whole system. So we perform the simulations in order to predict the basic quantity which determines how electric field interacts with the biomatter, and that's the complex permittivity. Basically, by tracking the autocorrelation function of dipole moment, we are able to predict the permittivity, and from that even absorption of electromagnetic waves in a biomolecular system, as long as we know what's the underlying structure. So this gives us a lot of ideas if our simulations are correct, because we can then compare them to the experimental data of the permittivity of water-based systems and biomolecular systems. In order to probe this basic electromagnetic material quantity of biosystems, the complex permittivity, we also develop special chips which enable us to measure the permittivity from very small volumes, which is otherwise not commercially possible, because typical devices for measuring permittivity, they require a few milliliters, which is unattainable for precious materials as isolated proteins are. So, for example, these can be not only used for measuring the permittivity as such, but also these can be used for sensing. Recently, we showed that using microwave microfluidics, we are able to determine the self-assembly state of microtubules, basically to say if the tubulin is self-assembled to microtubules or not. Furthermore, we also explore the response of biomolecular systems to electromagnetic field in terms of how electric field affects the structure, and then potentially also the function of the biosystems on the biomolecular level. So what you can see here is molecular dynamic simulations, all atom molecular dynamic simulations. I'm not showing water and ions, which are in the simulations. For the clarity, only protein is shown. So this is what you can see is a tubulin with its dipole moment, and when it's exposed to electric field, within few nanoseconds, the protein not only orients its dipole moment with the electric field, but also it changes its conformation of the important C-terminus part of the tubulin. This is one of the effects we can observe. On the larger structures, we recently, just a few weeks ago, our paper appeared published where we demonstrated that the whole microtubule can be affected by electric field. Basically, the electric field affects the balance between the dipole interactions and cohesive forces in microtubule wall, which causes the opening of microtubule. So these predictions guide us then for our experimental work. And in order to verify them, we develop the new technology for, if needed, and to address the new questions. And it's mostly based on chips which enable, on the one hand, delivery of very short and very intense electric fields, while being able to monitor the biological samples, be it microtubules or cells, on advanced microscopes. So, for example, we recently developed this chip which enables us to monitor how the microtubules are influenced by the very short electric pulses on a total internal reflectance fluorescence microscopy, TIRF. Recently also we developed a platform which enables us to probe the effects of nanosecond electric pulses on a cell cytoskeleton, which you can see here. On the left is a control cell with the microtubule comets labeled. And on the right, you can see how the cell cytoskeleton looks like after the treatment. The cell is not dead, but we remodeled the cytoskeleton by the electric pulses. Our other experiments, which I think are, I would say, I would call them the most important discovery in our recent years from our lab, were focused on affecting the tubulin structure. And we did that again, we did that with nanosecond electric pulses. We found that the tubulin itself is temporarily affected in terms of changes of the structure. We probed it with fluorescence, zeta potential, and other techniques. And interestingly, these electric pulses treated tubulin change either reversibly or irreversibly its capability to form microtubule structures. So the treated tubulin tend to develop different structures than the classical microtubules. So we believe that this is important not only in potentially some biomedical treatment protocols where microtubules are important, but also for nanotechnology. Basically, we demonstrated that nanosecond electric pulses can modulate the capability of a self-assembly of molecular blocks to the larger structures. Furthermore, we also showed that nanosecond electric pulses are able to remodel cellular skeleton. And again, this depends on the conditions of the cells where cells are placed in, and or this change can be also reversible or irreversible depending on the conditions. I must say here, particularly because I've seen Rod Connor being here, that Rod was the one who actually inspired us to use nanosecond electric pulses and microtubule systems. So thanks, Rod, a lot for bringing us in this interesting track of research. So the second part, which will be today the larger one, is focus on hypotheses and mechanisms and experiments which prove that biological systems generate electromagnetic field in various parts of the electromagnetic spectrum. Well, myself being an engineer, I'm of course practically I try to be practically minded person. So a part of having the pure of interest and aim of electromagnetic field being generated in organisms, that's interesting itself, right? Having something like an aura being produced by metabolism. I try to see what could be potential application of these phenomena. So what I believe, this is the Star Trek vision, which I mentioned earlier, that if we are able to detect electromagnetic field, which is going, of course, the non-trivial one, that which goes about thermal level, of intensity about the thermal level, then we believe that when we can detect it, we can analyze and interpret it. This could be a way for a new, completely non-invasive potential diagnostic procedures. So what is currently known about, in general, broadly speaking, about electromagnetic fields electromagnetic activity of living cells? So what you can find in textbooks is mostly classical electrophysiology. So it's known that certain cell types and tissues are electrically active. But when we look on it from a spectral perspective, which I do as a originally electrical engineer and a biophysicist, most of these signals reach 10 kilohertz maximum in spectral perspective. So this is what is known, but well accepted in the broad community in biophysics and biomedical engineering. Then on the other part of the spectrum, There are particular organisms which develop very well luminescence so strong enough that they're so strong that we can see it by naked eye. We call them bioluminescent organisms. Well, these species are quite many, but this phenomenon is not considered to be general. However, there is another phenomenon which I will speak about, which people use a different term for it, either biophotons or ultraviolet photon emission or endogenous chemiluminescence, which is ubiquitous and is present in every organism, provided it is having an active metabolism with oxygen. So this is another interesting part I will talk about. Then there is a gap, which is a gap, let's say, in the radio frequency, microwave, and a terahertz band, where there are several, well, I would say many hypotheses of potential mechanisms which could generate electromagnetic field in the cells. But these are still rather unexplored, although they have some history, a few tens of years. I guess they are mostly, what is missing here are appropriate experimental tools to address. So in this band, we are not sure, we don't have let's say, reliable mechanisms to detect electromagnetic field from the cells above the thermal level, to my understanding and knowledge. So I will bring you now through one conceptual framework which was developed originally and partially to a large extent to my PhD supervisor, Jiří Pokorny, which you might be aware. And he basically was the pioneer of, well, together I would say Fröhlich, of idea that vibrations of electrically polar structures could actually generate electromagnetic field in cells. So people proposed earlier in the 60s, 70s, 80s, it could be vibrating membranes of the cells. And Jiří Pokorny was probably the first one who have published papers and on that microtubules could be other potential candidates for this mechanism of electromagnetic field generation. Well, for anything of these happening beyond the thermal level, about the thermal level, so being non-trivial and interesting, or having potential, probably some biophysical importance and maybe be useful in potential diagnostics for detecting it, well, there needs to be a mechanism for energy supply to excite these vibrations. So there have been several proposals. I will go over them later on. And altogether, this framework is saying that provided that the energy, there is enough energy to, and these vibrations are excited, these structures should be generating electric fluctuations, which are very local and localized, non-localized in the near field of the structure, somewhere in the kilohertz and terahertz band of the electromagnetic field. So in the broad band, well, it depends on the particle structures. There is a huge question mark, it's particularly for me, and that's where I had the most of the discussions with Jiří Pokorny, if and under what conditions these vibrations, if they're really vibrations, and as we understand it, like mechanical vibration on nanoscopic level, how they can be not damped when they are in the watery environment. So this is kind of analogy of a problem of quantum biology that, well, skeptical quantum biology people would say, well, there is too much decoherence to something interesting to take place. So here there is similar problem. We have probably, well, it's hard to understand how we can avoid the damping of these structures in order to have like significant phenomena taking place. There are some hypotheses though that particle structure of the water near the protein and part of microtubules could be one of the phenomena which could lower the damping and then increase the coherence of these vibrations. But this is still with a question mark. Furthermore, Jiří Pokorny also kind of translated the idea of Fröhlich that nonlinear transformation could be another way how to overcome issue of the damping and potentially condensing energy into particular modes. And the idea from Jiří Pokorny was that it could be the strong electrostatic field from mitochondria, which are often co-located with microtubules, which could induce this physical nonlinear transformation of the energy and increasing potentially the vibrational coherence. But this is still very speculative, but it's interesting framework to my opinion. So then there are other potential phenomenon of free ionic oscillations or electron polaron oscillations of DNA or proteins, which could be potentially underlying substrate for electrical oscillations in high frequencies in cells. But there is, at least to me, I do not know what could be the energy supply mechanism for these. On the right part of the spectrum, and I'm speaking about in the frequency terms, so meaning the optical band, it is, I mean, at least to the most of the community, it's well known that there is indigenous production of electron excited states in biological systems, which generates at least near infrared and visible luminescence, so light above the thermal level. And what is so far well proved that the energy for excitation of the electrons comes from the oxidative reactions. At least there could be other mechanisms for energy supply, but this one is well, I would say without doubt. So if you want to learn more about this, I can suggest you either one of the papers from Jiří Pokorny, where he basically gives this integrative perspective, or in a larger extent in a book which we published some years ago, Fields of the Cell, with some of my colleagues, and it's open access. Just Google for it; you'll find that. Or I can also suggest in something concise, I would say also more critical, maybe a little bit more skeptical, a view on what is the current state of the art of acceptance of various mechanisms and theories when you look into all the literature and how the community perceive that. And that's in our paper, which was the first author is Ondřej Kučera. And there, what is interesting that many of mechanisms which are proposed to generate electromagnetic field or electric fluctuations in a microwave and RF and terahertz band are based on protein structures. So that's another reason why we like to work with the proteins, particularly tubulin and microtubules. So I will touch that a little bit on that. So in our recent past work, we developed several aspects of the hypothesis that microtubules could generate microwave fluctuations in the cells. I mean, microwave in terms of frequencies, I mean gigahertz. So the general idea is that, you know, I love this general concept of eigenmodes, of intrinsic frequencies of objects. So because it's generally valid. You can have eigenfrequencies or intrinsic frequencies of large structures such as bridges or buildings or planetary bodies, but it goes down to the nanoscale, molecular scale. So the same is true for the molecules. So basically, the frequency of eigenmotion of the intrinsic motion is determined by the size and mechanical properties of these structures. So basically, this can cover the broad spectrum. So we had a look on that in some years ago. Actually, my colleagues Ondřej Kučera and Daniel Havelka, and they wrote a nice review paper on that, how different structures, biological structures based on their sizes, could have potential vibrations in a particular frequency bands. So now regarding this is actually, well, this is one of the ways how we perceive the microtubule mechanisms of generation of electromagnetic field based on microtubule vibrations. So what about the energy which I mentioned? So the few theories which we, Jiří Pokorny and later myself, we contributed to, one of them proposed that the dynamic instability, particularly the GTP hydrolysis in a treadmilling and in a dynamic instability of microtubules, could be pumping the energy to the microtubule. So when we make an estimate, it's roughly like average physicist cell is roughly 10 to minus 18 watts per whole cell through these mechanisms could be pumped to microtubules. Furthermore, we also look on the traffic of the motor proteins, which again we also use in turn ATP hydrolysis to power their motion. And since they are closely attached to microtubules, this could be another source how energy could be pumped to the microtubules. But this is all very conceptual and vague. We are not speaking about particular energy mechanism transfer. It's more about the overall energetic balance. However, the third part, which was developed together with Jiří Pokorny, is particularly interesting in terms of magnitude. It looks like you know that mitochondria are the powerhouses of the cells, and when they're active, they generate plenty of energy and they lose plenty of energy. That's at least the classical perspective from a bioenergetics, that energy is lost because, well, this chemical energy generator or transformer is not perfectly efficient. So it's fact that indeed the mitochondria are roughly in average cell, again, it can be broadly varying, but roughly it's 10 to minus 14 watts in the wasted energy from mitochondria, which can be different forms in heat and also in luminescence and excited state generation. What I like, because it's more mechanistic, is interesting proposal which was published in Nature some years ago, which is related to the release of energy in enzymes. So it was shown that in exergonic enzymatic reactions, there is local nanoscopic acoustic wave generated at the catalytic site of protein. And I see there is a connection of this phenomenon to what could be happening in microtubules during the GTP hydrolysis, because it's also tubulin is kind of enzyme which catalyzes GTP hydrolysis, so it's conceptually similar. But this is again maybe why there could be a distant connection, but it's something interesting to think about. So would we ourselves done, assuming that there would be some excitations of microtubules, we in several papers we analyzed. You can see on the left there is a video of electric field wave packet propagating along microtubules. So the animation which you see is actually in the time scales of a sub-nanosecond, depending on the microtubule length. So we proposed that provided there are several fuse most excited on microtubules, their electric field wave packet propagates along microtubules, which speculatively could have implications for some very fast intracellular signaling. Furthermore, we made atomistic simulations of a microtubule, well, of course, grained simulations on microtubule vibration modes, which you can see on the right. There are static pictures here just to appreciate the shape of the modes. And there we show that depending on the microtubule size and properties, microtubule is having its fundamental modes in a microwave band. So this is interesting. So when now taking this all together, we also had a look on a whole cytoskeleton network, and we in our models we calculated what would be these spectra of these vibrations, be it either indigenously excited or maybe it could be excited by external means, by some laser pulses or electric pulses from outside by technological means. And there, depending on a damping as a parameter, we showed how the spectra could look like. That's the spectral image on the right down. Now going to the main topic of today. So this actually hooked me and got me to the science. When I learned from certain sources journals, I would say, that all organisms glow, I said, well, that's amazing. I really have to do research on this. So that brought me to the laboratory of Fritz Pop, where I also met I think it's seen David Dressen here. So I'm glad I'm seeing you here again. We met in the lab in the noise. So I made my master thesis in Fritz Albert Pop laboratory in Neuss at that time at International Institute of Biophysics. And at that time, my master thesis was supervised and mentored by Roland Van Wyk, because that time I liked to work on model systems, organisms being humans. So I did the thesis because they worked at the time on a human biophoton emission. I enjoyed their efforts. So what is this phenomenon? Well, at first, myself being more on the skeptical side of some speculations, I tell you for granted, this is perfectly proven phenomenon. There's nothing doubtful about that. What we know so far, and I will give glimpse about the terminology. I prefer to call this phenomenon biological autoluminescence for certain reasons. We trying to avoid the term biophotons because it was strongly discredited in certain countries. And that's why if you don't want to ruin the funding in particular countries, rather, I suggest to be careful using that term. But people call it different ways depending on their background. So it's called either ultraviolet photon emission, biological autoluminescence, endogenous chemiluminescence, or weak biochemiluminescence, different words. Depending on perspective, how the authors perceive the phenomenon. What is sure for now, and that's where the main paradigm stands in my perspective, is that this biological autoluminescence is the luminescence from biosystems where electron excited species are formed due to the energy from chemical reactions, particularly oxidation of endogenous biomolecules. So this is how we define it currently. I'm not excluding other sources, but at least know for sure this is the dominant source and mostly explored energetical source for this luminescent phenomenon. So basically it means life generates light even without being exposed to external sources of physical energy. So we don't have to expose cells or biomolecules to external light or any other stimuli. Just if their metabolism is taking place, there will be an oxygen is available. I will tell later on. Then this biological autoluminescence will take place. Now there are some There are some pitfalls though in detection. I will mention that later on. So spectrally, we are sure at least what is proved directly in the biological systems, not on the chemical system, is at least 350 to 700 nanometers. There's, well, I will touch on that later on. It's probably also a broader range, definitely going further to the near infrared. The characteristic also is the low intensity. And to give you very briefly the basic idea, as I mentioned, what we think is the basic generating mechanism is that there is a biomolecule, be it B, in the reaction, and some oxidant. I'm skipping many steps. That leads in dark, even if there's external light, just by the chemical transformations to generation of molecule, which has electron in an excited state, in electronic excited state. Then, well, there is probability that this excited state can decay by different pathways, and one of them is decaying by emitting a photon. So this is the as a basic I can give it physically. So in terms of intensity, just give you a perspective. So this phenomenon is biological autoluminescence, which I will call for short as BAL, BAL, is too weak to be seen by naked eye. I'm not assuming any, magical capabilities of certain persons to see very weak light, but I'm speaking about ordinary persons with very well dark accommodated eyes. You cannot see it. I tried in darkroom many times, and many other colleagues did. So you can see other phenomena in their light intensity. Just to give you a perspective, be aware this is a logarithmic scale. Choose your favorite unit. Give you an idea how weak is that. Imagine that on a International Space Station, somebody lights up a lighter or a candle that gives roughly 10 to 15 photons in visible band per second per centimeter squared. So now imagine you are trying to detect the candle from the International Space Station on Earth, on a surface. So that will be how many photons you're trying to catch. So detecting biological autoluminescence is like measuring the candlelight from the orbit. It sounds impossible, but it's not, because we can make in our experiments all the surroundings really, really dark compared to darkness which we have in Earth now. So that will be not possible for detecting candlelight from International Space Station. So this is measurable. And how? So typical working horse, also in our lab, but also in other labs, are either imaging detectors or integrative detectors. So, well, if you have object you want to detect the light from, well, of course you can use either light sensitive, very sensitive cameras. Typically, people either use EMCCD or some intensified imagers, and of course, the advantages is spatial resolution and also much higher quantum efficiency. But typically, one needs to integrate the signal for quite a long time. Well, depends on optics a lot. But if you are speaking about macroscopic images, this image of the hands, shining hands, can take 15 to half an hour, 15 minutes to half an hour. Much easier and cheaper is to work with integrative light detectors, so mostly photomultipliers, which have much higher temporal resolution because they need only few counts to get a signal about the noise threshold. Well, they don't typically provide spatial resolution apart from some particle types. Also, they typically have lower quantum efficiency, but you can then detect the kinetics of this phenomenon much easier. Now, a little bit going to the chemistry. It's only chemistry, I would slide which I'm showing, but I think for those chemists around you might be interested. So the underlying phenomena which leads to generation of this light are based, as far as we understood, in oxidative reactions. So during the ordinary metabolism in organisms, and particularly when there is increased stress, there is a endogenous generation of reactive oxygen species. This is a broad term. I'm not going into the details, but those can be hydrogen peroxide and other hydroxy radicals, and they kind of attack the biomolecules. Biomolecules here are basically, for simplicity, labeled as air, R, and here just to show there is a hydrogen on the end because something has happened there. So what happens then, reactive oxygen species, sometimes of them, they can abstract whole hydrogen, means oxidize the biomolecule, creating radical. Then in the consequent reactions, there are further radicals and peroxides of the biomolecules created. And well, there are some also back mechanisms taking place. This is the first basic biochemistry. Then, and what's important and specific for this light phenomenon is we are looking on the pathways which lead to intermediates, which are of high energy and of rather low stability. These particular are so-called dioxetans. These are the tetraoxides. Then these intermediates, they can be prepared, also synthesized chemically for simple molecules. They then spontaneously decompose, and they produce high energy in this decomposition, in this quantum chemical step, which is enough in particular energy manifold for particle molecules to kick the electron to the excited level. So this is where energy comes from. So then this is the mechanism how the primary electronic excited states of molecules are generated. Mostly people refer to carbonyl or aldehyde groups. And it seems to be that their primary generated ones are those which are at least explored are the triplet and the triplet excited state. And of course, they can transfer energy to different acceptors, which can then further emit light, or they can, they can also further react. There are many pathways, and only a few of them, a few of them leads to the emission of light. So I must say that I'm not expert in this biochemistry. It's my colleagues from Olomouc, from the other university in our country. We had a common project on this, and they are continuing the research in biochemical and biophysical mechanisms of light generation. So I can refer you to this very nice article in Biomolecules by Pavel Pospíšil. Just to summarize the emitters, what is known so far is the upright font. What is a little bit with question mark is in the italics. So these, you can see the spectral bands of these emitters. These are typically probed experimentally, but also there is also quantum chemical modeling behind some, not all, of these spectral bands. Now, this is something which we haven't published, but I'm really looking forward to having that published, because it really gives a lot of insight. And some people address this question of how many photons are there generated, how many excited states are generated. So this is a very difficult question, but very exciting and interesting. So I will bring you through simplified linearized process of generation of excited states and photons, just to give you an idea of the orders of magnitude. So if you go to the literature, you can find how much oxygen is being consumed by cells. It depends on the cell types, broad variety, but let's consider for simplicity, again, a simple spherical physicist cell to estimate that. So this is roughly how many molecules are consumed in active metabolically active cell. Then fraction of the oxygen goes to the production of reactive oxygen species. So roughly this yield, depending on our cell state again. Then a large fraction of these oxygen species is scavenged by antioxidants, so it doesn't really react with biomolecules. Those which do react through these mechanisms I showed you earlier, this reaction scheme, they can lead these pathways can lead to generation of these intermediates, which I mentioned. You can see now the rough estimate yields and rough estimate production per unit time in the cells. Now, this leads us to estimates of how many excited states are produced there per cell per unit time, and then leads us to how many photons per cell per unit time are produced. So basically, this estimates, and now I just tell you, the first part of this can be directly measured. There are methods for that. This is a black box, or very difficult to assess, and this can be directly measured. We know how many cells we have. We see our medium is transparent, so is there no self-absorption directly in bulk? And we measure the photons, and we know the quantum efficiency. So this can be measured. So we are quite sure about this. So it means basically these estimates, and also this is confirmed by, at least this input output is confirmed by experiment, is that cells generate in a whole visible band, at least visible band, roughly like 1 photon per 15 minutes. That's a little, right? So how to detect it at all? So how we trick that in order to have some research which is doable, we simply have a lot of cells. So typically in our systems, we have hundreds of thousands or millions of the cells per milliliter. And that, provided they're metabolically active, gives enough signal to detect hundreds or thousands of photons. Now, just give you very briefly. So I just give you a few examples. We have several experimental systems what we have in our lab is was actually inspired by Cristiano Galep from Unicamp from Brazil, whom I visited as a postdoc. And he showed me actually a very simple and very effective way and rather affordable way how to build the systems to detect very weak photon emissions. So basically relies on what most of our systems are relying on photomultiplier modules, which already integrate the amplifiers and also the shape, the pulse shapers. And then it's basically a plug and play, and we even don't have to cool this PMT, particularly this one, because it has low red sensitivity, so also low noise in room temperature, and we can detect directly well, of course, the light tightness of the chambers is absolutely essential, but roughly this is how our systems look like. We also include filter wheels in order to detect the spectra, but also we can use different ways to do that. So I will tell you now what we found in our research, let's say in the past roughly 10 years in this research field. So we confirmed, because we're not the first to show that this biological luminescence is related to the growth and also to the metabolism. What you can see here is a signal from the flask of roughly 150 milliliters of yeast cells, which are dividing, growing happily. And you can see at the same time we measure the concentration of the cells. So we can see that in the beginning the cell, the cell intensity, the cell number grows, of course, and also the photon emission from the cell culture grows. Then at certain point, intensity drops, and we believe it's related to deoxic shift, so the change of the metabolism. And we kind of proved that, or suggested that it could be related to the metabolism shift, that if we replenish the glucose, which we know was depleted in this dropping point, this photon emission again recovers transiently until the glucose is again basically consumed by the cells. We also demonstrated, this was in collaboration with our colleagues from Netherlands, from University of Leiden, from Metabolomic Center, that this luminescence is related to oxidative metabolism, where we basically correlated the production of metabolites in human leukemia cells with the photon emission detected in parallel. And there we found a positive and negative correlation with particular metabolites in these methods. We found in our lab, although that, again, this is confirmation on a particular system, a new system, but people showed it earlier as well, that biological autoluminescence is its intensity, it's suppressed by antioxidants, which again confirms that the oxidative processes are important for generation of this light. So for example, we showed either at human leukemia cells, which can be triggered to start to produce reactive oxygen species massively as a part of the immune reaction. These signals, these endogenous light signals, can be decreased by intensity when we apply antioxidants, and similar in the case of these cells. When we apply, again, it's different types of antioxidant here, ascorbic acid, vitamin C, then the signal drops. If we apply on a small concentration, then the signal can recover because the antioxidant is depleted within a certain time. Furthermore, we showed on, again, of different organisms that biological autoluminescence is enhanced by oxidants. So for example, here you can see very small, many hundreds of germinating plants. It's a model plant, Arabidopsis thaliana. You can see that when we drop, actually we sprayed hydrogen peroxide, either, only the vehicle, there was water in this case, or the hydrogen peroxide, we can see that the signal is enhanced. Basically, by the imaging, we could see the signal is stronger. Similarly, in yeast cells, with the photomultiplier detection, we show that the more oxidant we add, the stronger the signal gets, and then it decays to the sub-basic level, the spontaneous signal. I particularly love imaging of imaging from human body, because it's something which we can understand. It's interesting. So although the images are noisy, I guess you can see. So on the left, we have a black and white image. On the right, this is the endogenous signal, which constantly takes place. So just remember, there's no light excitation. You can be there for hours and days, and as long as you are living and there is oxygen, you will shine. So what you can see on the right is this spontaneous signal from the human skin. And this is again demonstrated that topically applying antioxidants increases photoemission. So basically, this method as an imaging method is showing us unseen, because you would not see anything even while applying lower oxidative oxidants, or basically if there is some disease process in the skin which will be leading to oxidative stress, you could see that by this method even before there is some manifestation in, let's coloring of the skin or something like that. Now I'm going to in this imaging part I will show you some nice images from labs from our collaborators around the world. So Professor Kobayashi from Sendai in Japan, he is in that lab, in his mentor, Professor Inaba, was the very first one who in 1989 made an image of the biphoton emission, as they call it that time. And this is not this one, this is a newer image. So they are really pioneers in imaging in this weak luminescent phenomena. What you can see here is rather new work from their lab, which shows that growth of the implanted tumor in the naked mouse under the skin can be is reflected by this photon emission. So as the tumor grows, you can see weeks here after the transplantation, you can see that as tumor grows, there is a very active metabolism, and that brings about strong light generation. And this is as the tumor progresses and the tissue goes to necrosis, then the light ceases. So you can only basically then it screens the light which would be coming from the deeper section. So you can only see on the perimeter of the tumor there is still visible light emission. Interestingly, there's a very interesting work from China, from Jiapei Dai, who's shown in a couple of papers that when the metabolism in neural tissue is stimulated, they use different glutamates here, it brings about higher photon emission. This is not surprising because stimulating metabolism by different ways, if it leads to increase production of ATP, that means also you need more higher activity of mitochondria. It will also bring more reactivation of species. So it's not surprising we are seeing that just on basal this biochemical presumption. So this is interesting to see on the brain slices how this photon emission evolves over time when the glutamate is dropped in particular section of the brain slice. I would say the first systematic imaging of the human photon emission patterns was done by around 1 week. Whom I very value because he was my first mentor in this field, my master thesis supervisor, and together with his son Edward Van Wyk, they published plenty of interesting papers on this topic. So what you can see here is actually the Roland Van Wyk himself on the black and white photos on the right, and this luminescence image, biological and luminescence images on the left. So you can see there is particular pattern which we can discuss later on. Why is it in these particular places? Well, a lot of interesting work was done again in the lab of Kobayashi in Japan on this topic. So it's interesting to compare this, as you call it, biophoton emission of ultraviolet photon emission. I prefer to call it, again, say, BAL. If you compare it to infrared emission, which reflects only the surface temperature and emissivity, you can see there is some correlation, but very weak. Well, obviously, because biological luminescence doesn't reflect temperature per se, only indirectly. It does reflect the intensity of oxidative reactions taking place in optically accessible parts of the object. Here is the human skin. Interesting work was also done, as I mentioned, by Van Wyk. They show there is a particular pattern which is rather consistent among persons, of where there is a stronger light emission at particular parts of the human body. So this kind of pattern of light emission from the human body, which can be objectively detected, which I consider fascinating, right? Now I will bring back us to the work which we've been doing in our lab. And of course, we've been asking the questions, what is the color of this light? It's not that easy to detect it directly because, if you want to detect color, you have to filter out so your signal gets even weaker. So anyway, we did mostly with the filters, we did some analysis on, and we showed that, at least we demonstrated in this simple case, we compared the spectrum of the yeast cells culture and the human leukemia cells culture, and we could show that there's light emission spectra differ. So I'm doing full stop now for this, but basically it gives us hope that in the future maybe we could distinguish state of the organism, not only type but also the state, potentially distinguishing healthy and disease states by the color of this photon emission, the spectrum. In order to give more insight, I believe it's very important, and we just briefly touched it in a collaboration with our Iranian colleagues, particularly with Homa Saidefrezeh, where we asked the question, so because we couldn't find any answer in the literature, if people did any quantum chemical simulations of potential emitters, people say it's triple carbonyls, but did anybody ever calculated that in quantum chemically? What are the spectra? So actually nobody really did. So we did that for the first time. Now we are preparing a paper. So far it's only the chapter in the book we are preparing. So we could show that on the simplest carbonyls, so practically aldehydes and small aldehydes and acetone, what is the spectrum of the triplet excited states when it translates back to the ground state, the singlet, which is, well, typically forbidden. That's why it's a bit tricky to calculate it quantum chemically. And we compare different quantum chemical methods, and we found which one is the most optimal in comparison to the experimental data. So I think it's important. These books are important in order to give insight into the really quantum chemical and quantum physical mechanisms of this photon emission, to understand the spectra of the emitters and how they could interact potentially. Now, this is one topic which I was really hooked into in the very first project we get on this biological autoluminescence. We called that photonic biosignals at that time, because we were really curious if there is any message which this light carries, and if you can use it for potentially deciphering it, if it could bring us any diagnostic value. And this is a really tough topic, but very interesting. I give you just a little bit insights into that, to what we found. So the working hypothesis was that, well, people did in the past ask questions. So is there any particular state of the light field, quantum opticians know what I'm speaking about, which would be bringing out particular photocount distributions? Well, if you measure some photocount distributions just, by photomultiplier and its light is weak, how to interpret it correctly? So we've been a bit careful about bringing in quantum optical mechanisms, because I'm more inclined to believe it's, well, basically the physical chemistry and kinetics which determines the photocounts statistics. But so therefore we took a different approach. The hypothesis, which I still believe is valid and interesting to follow, is that because the intracellular environment, particularly proteins, organelles, what you can have in the cells, their surfaces are structured, and you can describe them by the fractal geometry. So once you have physical geometrical settings where the alchemical reactions take place, which is fractal. So we believe, and this is the logical step which is important. I'm not sure if it's perfectly valid all the time, but I think it's hypothesis that in the fractal environment, if the chemical interactions take place, they could lead to fractal time series of how on the kinetics of the chemical reaction. So this is what we pursued. So at first we addressed some of earlier theories by Fritz Popp and by others who coined the term biophotons, that they propose that well, this endogenous emission comes from a particular quantum states of let's say quantum field in the cells. It's very interesting. I really love that idea, but I don't think we have a proof for that. The problem is that it's very hard to interpret the data. So if you are seeing, now speaking to quantum opticians, if you are seeing sub-Poissonian or super-Poissonian statistics, it doesn't mean it necessarily comes from the state of the quantum state of the field. It can be, and often is the case, can be caused by non-stationarity, which can be caused either by detectors, of course, you have to be careful about it, but also by the chemistry itself. So we have to be careful about interpreting that. So we have a quite long paper on this of how we stand on the quantum optical approaches and what we think is a better way to analyze the statistics of the photon counts. So what we did, we took different approaches. We had a look on entropy in the signals. In this paper by Homa Saidefrezeh, we found that on the first look, it looks like there is lower entropy in these biological signals than in the noise. However, this is just the side effect, and it's simply the trivial result of the different signal to noise ratio. So it doesn't, when we made proton control and computer generated signals, we found that this is actually not, this will be this is a false positive. So in the end, we found out that really we don't see different entropy in the signals of, biological signals, the real experimental ones, and then which we generate by computer. In our very last work on this topic, however, we made really the best what we could to exclude any artifacts, and we did very rigorous statistical testing. This was the first time where we found, and I believe that at least to a certain extent, there is something interesting there. It looks that the Hurst exponent is slightly lower than the reference computer-generated signals, and which are combined with the dark noise of the detector to make a good control. And there we seem to see there a slight anti-persistent behavior in the signal, which we can think is because of the subdiffusion properties of the biochemistry underlying. Okay, now approaching on the last parts, and again, the later part and an even more exciting topic. So, which actually also got me hooked some years ago. So, can this light be used by organisms to communicate? So, is there any light-based biocommunication based on these light signals? So the idea was, and we are not first to address it actually, this is how this field even started, and by Alexander Gurevich in 1920s. The idea was that cells exchange certain physical signals, and they were hypothesized to be of the photonic or electromagnetic nature. And these signals are basically were claimed to be proved if they could cross a barrier which chemistry couldn't cross. So basically, it's a barrier which is physical, so the chemicals cannot cross, only physical phenomena can. So this was the basic idea. So people elaborated that over the time, particularly there have been very interesting research in former Soviet Union, where by Kaznachev, who wrote a couple of books on that, of these 10,000 experiments he carried out. And there, the idea was that let's have two cell cultures which are separated so that the chemistry cannot cross. And on one hand, there is so-called inducer culture, which is typically stressed by different means or even killed by some drugs or poisons. And then there is chemically untouched detector cell culture. And there we're basically looking on, if we stress the inducer one, does this effect somehow transfer non-chemically to the detector one? So this is actually the core idea of many experiments which you can find. And there are, if you go to not only my review, but also other reviews with other people, you will roughly find there are, let's say, at least in English, there are roughly at least 30 papers on this topic, which show some interesting data. And if you speak Russian, you can go to the books where we found 10,000 of experiments of different types, which were carried out in the 70s and 80s. So anyway, there's a major problem for us as physicists and engineers, how this could work if there is the signal is so low, right? How could this work in a light environment that you have? Even in the deep of the organism, still light penetrates quite deep. So even when you cannot see it by naked eye, it doesn't mean it's dark. It's still a lot of light. So if Andre could share out in our paper, we made very simple assessment of, in terms basically in communication theory, what will be amount of bits which can be transferred based on a background. So let's say if there is some averaging mechanisms which could enhance the signal to noise ratio, we try to address how dark it has to be in order to transfer the information by photons in classical terms. The quantum aspects may be very important, but this is something which is, again, very interesting, but still very speculative. So I will outline the perspectives of this research field. This will be my few last slides. So what I think is perspective, at least from perspective of our research team, is to find the ways how to enhance biological luminescence for different purposes, to kind of modulate and manipulate it so we can understand it better. One of the ways which we are going currently is using intense electric pulses. You see, it connects to the earlier topic which I described. So idea is that we know that the oxidation of biomolecules leads to biological luminescence. This is for sure. Furthermore, however, there is data which shows that electric pulses cause oxidation, either by stressing the cells, and that electric pulses can stress the cells so they increase their ROS production, or electric pulses directly by applying potential to the electrolyte can generate radicals by electrochemistry directly. So both these pathways can be leading to this effect. of basically modulating electrically the biological autoluminescence signals. Or vice versa, we can invert the story and using the biological autoluminescence to indirectly monitor the effects of electric pulses. So we've collaboration with Louis Mir from from France some years ago. We still haven't published the data in journal paper. We show that indeed it depends on electric field parameters how and if these biological autoluminescence signals or endogenous chemiluminescence signals are increased or not. So you can see here how these signals differ. Let's say if you apply one short or one long electric pulse or train of short pulses. In this case, in these adherent cell cultures which we use for these experiments. Another way how to manipulate or enhance without maybe affecting chemistry that much biological autoluminescence using nanoparticles. I think very interesting idea would be to manipulate the excited states in order to change the radiative rates, and that in this way to increase probably the biological autoluminescence. So we did some first steps in that in collaboration with our colleagues in Iran. They synthesized liposomes which targeted mitochondria directly, and inside the liposomes carried nanoparticles. And there we showed in this experiment that when we compare to the control, this is just the endogenous signal from the cells. This is just the background of the detector or medium. If we apply liposomes with the nanoparticles, the signals is increased. So we demonstrated that in this, and we are not the first one, other people use nanoparticles too, but we were first time used targeted liposomes to the mitochondria. We showed that indeed nanoparticles can increase, in this case in osteosarcoma cells, these biological autoluminescence signals. So concluding this part, the take-home message is that we are sure that the biological autoluminescence is generated by the relaxation of electron-excited states of biomolecules generated endogenously in biosystems. And we are certain at least about one mechanism which provides energy, and that's the energy from oxidative reactions. So beyond the pure interest in the basic biophysics, I think it's the applications could lead to new biomedical diagnostic methods, maybe having a new fingerprints in signal properties. And this method is interesting because it's completely non-invasive. No energy even is input to the system. We are just looking on the system, and it's also rather real time and low operation cost, and it's completely labor free too. Before I conclude, and I'll be glad to take, I see quite many questions in the chat. I just giving you a bit some practical information. We are just this year we should be finishing a book on Springer, authored by these editors and other contributors, which will be covering different topics of quantum and physical chemistry of this phenomenon. It basically will be a book on biological autoluminescence, starting from basic quantum chemistry and physical chemistry to applications, also potential biological significance and history. So it will be interesting with some data on this topic. Please contact me or one of the editors, and maybe you could find a slot to particular topical areas there. Furthermore, I would like to make a little bit advertisement because we are organizing our team regular bioelectrodynamics webinar, which is addressing the topics of research interest of a broad research interest in the biophysics and electromagnetics. You can see our past speakers and the upcoming speaker next week. So if you would like to register, either drop me an email, or even easier, follow our social media of our research team, Twitter, LinkedIn, or Facebook, and you can find the registrations directly. Last but not least, since we have currently having a large project, we are now hiring a postdoc in molecular modeling. So if somebody or your colleague will be interested, I'll be glad to give you more information. We'll be opening the call in a few days. So with that, I thank you very much, and I hope you are enjoying this trip of this community of making science fiction we seen in the beginning a reality. And I will be glad to take any questions we might have. Thank you very much.